everyone, we are almost done with chapter 3, Gene Segregation and Interaction. So, tatlong topics na lang yung i-discuss ko before we finish this chapter. So, specifically, this will include the influence of the environment on expression of genes, as well as twin studies and probability and statistical testing. So, let's start our discussion. Okay, let's start first with environmental influence on gene expression, or simply, what is the influence of the environment on the expression of a particular phenotype given this genotype. So, yun yung ibig sabihin. And then, we have here an equation, P is equal to G plus E plus G by E. So, wherein P refers to the phenotype, G refers to the genotype, and E refers to the environment. So, yung phenotype, yun yung observable, measurable characteristic of an organism, which is brought about by the genotype and the environment. Yung genotype or the genetic composition, yun yung part ng equation na heritable. Ibig sabihin, this is passed on from parent to offspring. And then yung E, yung environment, anything that is not genetic is considered environmental factor. So example, food or nutrition. We also have biological sex. We also have age. We also have other factors such as temperature and so on. Yun yung environmental factors. So the combination of all of this, G and the E contribute to the expression of the phenotype of an organism. Parang dito sa diagram na ito. Even if, say for example, even if you have the genes for white complexion, kunwari, if your environment is not favorable for the expression of a white complexion, then you will not have a white complexion. So, dapat, kung white yung complexion mo and you have a favorable environment, for that to be expressed, then you have the white complexion. Another example would be IQ. Say for example, average yung IQ mo. But with environment, say for example, yung ginagawa mo, you practice and practice and practice, you learn. That is your environment. With average IQ plus with a very favorable environment, with a good environment, then you have a higher, you will tend to have higher scores in exams, in quizzes, recitations, and so on. Okay? So, yun yung another example of the environmental influence. So, what does these things imply now? Okay? So, a given genotype may give rise to different phenotypes at different environments. Okay? Certain genotypes that are adapted in one environment may not survive in another. Because of the variable interactions between a genotype and an environment, it is now easy to see that the genotype determines the range of phenotypic expression that may develop. Okay? Yun na yung tinatawag natin na range of reaction of the genotype. Okay? So, the phenotype now that will be realized depends on the environment in which development takes place. This means that one will not be able to know the entire range of reactions of a genotype because it is not possible to expose an individual to all possible kinds of environments. Okay, malalaman lang natin yung range of reaction of the genotype for a particular environment, 'di ba? For a particular defined environment. Okay, so let's proceed now to the definition of terms. So, ang discussion for this topic, for this environmental influence on gene expression will be divided into three. Una, definition of terms, and then external environment and the internal environment. So, let's start with the definition of terms. Ano ba yung tinatawag natin na penetrance? So, we have here a keyword or a term in genetics which is penetrance. Ano ba yung tinatawag natin na penetrance? This is actually the proportion of genotypes that show unexpected phenotype, or it is also the probability of a gene or trait being expressed. No? So it is a statistical concept of the regularity with which the gene is expressed. So we have here two types of penetrance. We have complete and incomplete. So complete penetrance, nasa-show lahat yung expression, yung phenotypic expression. Say, sa pag-determine kasi ng penetrance, if it is complete or incomplete, we consider the population. If all of the individuals in a population express or show the expected phenotype, then we have a complete penetrance. But if only some individuals, so few individuals lang yung nag-express ng expected phenotype, then that is incomplete penetrance, or this is sometimes called reduced penetrance. Okay? So, pare-pareho lang yung genotype ng individuals na ito, but not all of them express the expected phenotype for that particular genotype. Because, again, nasabi natin sa discussion natin a while ago that it is influenced by the environment. Okay? And then we have here expressivity. Ano naman yung tinatawag natin na expressivity? This is actually the degree to which a particular phenotypic effect is exhibited by the individual. Okay? So, na-express nga yung phenotype. So, ibig sabihin, the individuals in a population showed the expected phenotype of a particular genotype. But, to what extent? Yun yung tanong. Okay? To what degree is it expressed? Dito, narrow expressivity dito. Ibig sabihin, almost all of them. So, based from this figure, all of them showed or expressed the expected phenotype. That is narrow expressivity. But for the variable expressivity, the expression or the degree to which a particular phenotypic effect was exhibited by the individuals varied. Okay, same genotype, yung individuals na ito, same yung genotype, but their degree of expression differed. Yun yung variable expressivity. So, ano yung difference nila sa penetrance? Sa penetrance, hindi lahat ng population ay ina-express or pinapakita yung expected phenotype. Yun yung incomplete penetrance. Ang difference nila sa variable expressivity ay na-express yung phenotype. Lahat na sa population. Lahat ng individuals sa population in-express yung phenotype, but to a different degree. Okay? So I hope that is clear for penetrance and expressivity. So, doon naman tayo sa pleiotropy. So, actually, the word pleiotropy came from the Greek word pleion, meaning more, and tropos, meaning ways. So, many ways, parang ganon. And it is the effect of a certain genotype influencing more than one observable trait in the individual. It is also the phenomenon wherein a single allele influences more than one distinct trait. So, paano yun? Usually kasi, di ba, we have one gene, one trait. Usually, yung isang gene, iisang trait lang yung ini-influence, no? But, in pleiotropy, pwedeng yung isang gene, it can affect the expression of several traits. Yun yung tinatawag nating pleiotropy. And this pleiotropy can be a major cause of genetic correlations. Say for example, in poultry, say for example, we have a dual purpose type na chicken. So yung dual purpose na yun, either we want to have higher growth rate or either we want to have a higher egg production. So you choose which is which. Mas gusto mo ba yung mas mabigat na chicken but still produces an egg? Or mas gusto mo ba yung mas marami yung mangitlog? So, kapag pinili mo yung mas mabigat na manok for meat, ang tendency is that bababa yung egg production. But, pag pinili mo naman yung higher egg production, there's a tendency that growth rate will be slower. So, mas maliit in size yung yung manok kapag pinili mo yung higher egg production. And that is one example of genetic correlations, pleiotropy. So one gene, it affects several traits. Okay. So the last term that we will define before we proceed to external environment is Phenocapi. And this means an environmental mimic of gene action. Yung example natin dito is diabetes. Okay? So for example, we have an individual with a genotype coding for diabetes. Okay? But because we have an environmental influence, say for example, they are dependent on insulin, they will be phenotypically normal. Okay? Wala kang ma-observe na phenotypic characteristics nila that they are diabetic. Okay? And this environmental influence, yung environmental influence na yon, which is because of insulin, is sufficiently strong so that the resulting phenotype, which is normal, simulates the effect of certain genes. Okay, let's proceed with external environment. Under external environment, we have 4, temperature, light, nutrition, and maternal relations. By the way, these environmental and external environmental effects are those leading to phenotypic changes or changes in phenotypic expression that seem directly correlated with observable changes outside the organism. So let's start with temperature as an environmental factor. Okay, there is actually a close correlation, no, between the rate of chemical reactions and temperature. Since the basic developmental effect of many genes is to control the rate of a specific reaction, a change in temperature can be expected to have widespread effects on development. So we have here an example, no. It's actually a primrose, no, primrose flower with the scientific name Primula vulgaris. So yung red color nung flower na primrose at room temperature is red, no. So red color yun at room temperature. But when the temperature exceeds at over 30 degrees, no, the color of the flower is white. So, yun yung effect ng temperature sa phenotypic expression ng isang trait. So, pareho lang sila ng genotype, but because of environmental factors, the phenotypic expression differs. Okay? So, yun yung example for temperature. Next, we have light. So, ano naman yung ibig sabihin nito? Yung energy kasi provided by light, and light itself is essential for the growth and development of not only plants but also animals. But our example in here is plants. Specifically, this is actually a corn and this is an apple. Okay? So examples, seedlings grown in the dark. So yung seedling na grown in the dark, yung yellow yung kulay nung leaf, do not survive long because chlorophyll is not developed even though there are genes for chlorophyll development. So, pareho silang, these two plants here ay parehong merong genes for development of the chlorophyll. But because the other one has inadequate light, then it will not survive long as compared to the one that has enough or adequate light. Okay, so yun yung ibig sabihin. And then another example is here. So I took this picture when we went to Japan for a comprehensive international education program, no. In there, we were taught na importante palang tanggalin yung leaves na nakatakip sa apple fruit. Why? Because it requires light, no, sunlight, para maging red yung kulay ng apple. Okay? So, ganito yung mangyayari sa apple kapag hindi tinanggal yung leaf na nakakover sa fruit. So, di ba, yung consumers, they prefer to buy those red apples, no? Kung ganito yung magiging kulay, siguro walang masyadong bibili. So, it is a requirement to to give adequate light, no, for the fruit to have red peel. Red peel ba yun yung tawag? Okay, so yun yung example ng light as an external environmental factor. And then let's proceed to nutrition. Okay, we have an example here. It's an egg yolk color. Okay, so yung nutrition. What does nutrition play in the expression of a phenotype for a particular trait? Okay, food kasi or nutrition provides energy for carrying out necessary processes and materials that must be incorporated into necessary structures. So genetically, different organisms, even of the same species, usually differ in the kind and amount of nutrients they require. So, depending upon its genotype, simple dietary changes can lead to different phenotypes. So, ang example ko dito again is the color of egg yolk in chicken eggs. So, pareho yung genotype nila, no, for egg yolk color, but yung phenotype is different. So, yung isa, darker yellow or nearing to orange, and this one is lighter yellow. So, ano yung factor? Bakit ganun yung phenotype nila? Because of nutrition. Particularly, yung corn. Yung corn kasi, if you feed chickens with corn, they will tend to have a darker yellow for egg yolk color. So, mas darker yung egg yolk color nila na yellow. Okay? Kapag konti lang yung binigay mong corn sa kanila, relative to the normal na dapat na ibigay, lighter, no? Magiging pale yellow or lighter yellow yung egg yolk color. So, yun yung example for nutrition as an environmental factor. So, it affects the expression of this egg yolk color. Okay? Next, let's proceed to the last topic under external environment. We have here maternal relations. So, in the case of mammals, where the direct body relations between parent and progeny extend beyond fertilization, the interaction between the fetus and the maternal environment still occurs. Okay? So, ang example dito, we have blood type incompatibility between the mother and the offspring. And the blood type incompatibility could produce special effects on the survival of particular genotypes. So, say for example, during pregnancy, the blood type of the fetus and the mother are incompatible. May mga pangyayari or there are changes that affect the survival of the fetus. Okay? Or affect the survival of the developing baby. Why? Because there might be an interaction between the two blood types, that of the baby and that of the mother. Okay? So, maaaring ang baby won't survive or there might be complications that could occur. Okay? So, yun yung phenotypic changes, no, that happen because of blood type incompatibility. So, yun yung example or illustration for maternal relations. So, we are done with the external environment. Now, let's proceed to the internal environment. Okay, so yung internal environmental effects naman are those which lead to phenotypic changes that appear correlated primarily to changes within the organism. So, number one, we have here age. Okay, so as an individual grows older, there are certain phenotypic changes that occur which allows further genotypic effects to be expressed. So, say for example, age of onset of these different genetic traits in humans. Okay? So, yung, say for example, we have here alkaptonuria. Napapakita na siya even at birth. So, this is already expressed at birth. And then yung diabetes mellitus, this is usually expressed from age 40 to 60 years old, and so on and so forth. Yun lang yung ibig sabihin ng age. So, at different times of the lifespan, so at different periods of the lifespan of an individual, may mga na-express, no, na genetic traits. They are not readily expressed at birth. Some are expressed even at age 60 years old or even at age 30 years old. Parang sa pattern baldness. Diba, when an individual is still, say for example, 2 months old pa lang, hindi mo naman makikita na mayroong pattern baldness na, okay, sa phenotype niya. So, yun yung ibig sabihin ng age. Next, we have this one, biological sex. So, of course, we see different phenotypes of male and female because of the presence of their reproductive organs. May mga phenotype na makikita mo lang sa males, may mga phenotype na makikita mo lang sa females. To illustrate further, no, we have 3 sex-related traits. Number 1, we have sex-linked, sex-limited, and sex-influence. Yung sex-linked na trait, this is usually associated with the sex of the individual, okay? And this sex-limited is a trait wherein it is expressed in only one sex. Kunwari sa egg production, yung babae lang yung nangingitlog. Okay? Kahit yung genotype ng lalaki ay for egg production, hindi siya mangingitlog because wala naman siyang reproductive organ para mangitlog. Yun yung ibig sabihin. And then we also have here milk production, yung babae lang yung nangingitlog. Kahit pareho sila, male and female have the same genotype for milk production, if they do not have that reproductive organ to produce the milk, they will not produce milk, even if the genotype says so. Okay? And then we also have sex influence. So, pareho, say for example, this is pattern baldness, no? Actually, their genotype is heterozygous. Even if they have the same genotype, iba yung expression sa female, iba yung expression sa male. Okay? But they both express pattern baldness. So, yun naman yung sa sex influence. Okay, we have substrates. The kinds of reaction that take place in an organism or individual depend largely on the substrates present. And these substrates are synthesized by the organism, and their presence or absence may be genetically controlled, so that the phenotypic expression of a particular genotype will then depend on the substrates present. Okay, so I will not discuss this pathway, but this is just to illustrate how the substrates could affect the expression of the phenotype for a particular trait. Let's proceed to the second major topic for this video. We have twin studies. Now, why do we study this? Okay, so merong twin studies because this highlights or explains how the genotype by environment interaction could affect the expression of the phenotype of an organism for a particular trait. Why? Because not all characters are determined by simple genetic effects which have easily observable relationships with simple environmental changes. There are some traits kasi which are probably the results of complex G by E interactions that are measured as single traits. And because of the complexity of such traits, it is usually difficult to define the roles of heredity and environment in exact numerical terms. Okay? So that's why we have twin studies. And twin studies are generally undertaken using identical twins, fraternal twins, and non-twin siblings reared apart and reared together. Okay? So, ano yung tinatawag na identical twins? So, they are genetically alike. They are subjected to the same intrauterine environment. Yung fraternal twins naman, they are not genetically identical. But they are also subjected to the same environment. Same intrauterine environment. So, ano yung difference ng identical? Yung identical twins, pareho yung G, pareho yung E. Yung fraternal twins, hindi pareho yung G, pero pareho yung letter E. From the equation P is equal to G plus E. So, example, we have here a table that shows the genetic concordance among identical and fraternal twins. So, yung age of sitting up, yung percent concordance sa identical twins is 82. And sa fraternal twins, 70%, 76% lang. So, ano yung ibig sabihin? This is environmental. Yung age of sitting up depends on the E. Largely depends on the E. Next, we have age of walking, hair color, eye color, diabetes mellitus, rickets, feeble-mindedness. If you can see, medyo malayo, no? 68 and 31, 89 and 22, 99.6 and 28, and so on. As you can see here, very high in identical twins. That means, the identical twins kasi, same yung G and same yung E. Sa fraternal twins, different G but with the same E. So, ano yung remarks? Basic, these traits now, age of walking, hair color, eye color, diabetes mellitus, rickets, and feeble-mindedness, they are basically genetic, no? It largely depends on G or the genotype with high degree of penetrance and differential expressivity. Sa blood pressure and criminality, 63 and 36, 68 and 22, this is basically genetic with some environmental influence, di ba? So, because yung blood pressure, this could be affected by nutrition also and lifestyle. Kaya meron siyang remark na with environmental influence. So, sa criminality naman, hindi naman ibig sabihin na kapag mataas na yung criminality factor ng isang parent, mataas na rin yung criminality factor ng isang offspring. Kaya may remark dito na environmental influence. But this is basically genetic daw, sabi ng data. And so on and so forth. So, it is a presumption that the difference between these identical and fraternal twins is only in the extent of their genetic similarity. If phenotypic similarities are greater among identical twins than among fraternal twins, this may be ascribed to similar genotypes of the identical twins and the similar genotypes of the fraternal twins. On the other hand, if phenotypes are the same for both identical and fraternal twins, the environmental influence is greater than the influence of the genotype. And one of the ways to measure similarities and differences is to determine whether the character is present or absent in one or both members. If both members show the character, the pair is concordant. And if only one member of the pair of the twins show the trait, then the pair is phenotypically dissimilar or disconcordant. So, the extent of concordance of the twins may be used to measure the roles of the environment and heredity in the expression of a certain phenotype. If the trait has high concordance in the identical twins and quite low concordance in the fraternal twins, then the trait may be considered to have a strong hereditary element. Strong G, okay? More equal concordance and discordance ratios between the identical and fraternal groups would in turn signify less influence of the G and more influence of the E on the phenotypic expression of a particular trait. So, ano yung key takeaway dito sa concordance at saka discordance? The interpretation of concordance data must be done with caution because environmental effects on some characters may be subtle. However, the concordance data emphasize the important roles of both G and E in many human traits. Okay? We are already done with the first two topics for this video. Now, let's proceed to the third and last topic for chapter 3, and that is the probability and statistical testing. Why do we do probability and statistical testing? Because from this, we derive we determine the genetic and phenotypic ratios. So we compute, diba? If you remember, we compute for the phenotypic ratios, the genotypic ratios of a given mating or cross. Ano yung ginagamit natin to get these ratios? This is actually based on the principle of probability and statistical testing. So, ano yung laws of probability na ginagamit natin? So, yung laws na yon ay for independent events and mutually exclusive events. Ano ba yung tinatawag natin na independent events? So, these are events that occur independently of sa independent events, yung occurrence ng event na yon does not interfere with the occurrence of the other. So, it occurs independently of the other event. Yun yung tinatawag na independent events. And to get the probability of independent events, we get the product of their separate probabilities or their individual probabilities. Say for example here, we have a mating. This is actually a dihybrid cross. So we take each gene pair independently so that we have here heterozygous B, heterozygous R. The ratio, genotypic ratio is 1 homodominant, 2 heterozygous, and 1 homorecessive. And the phenotypic ratio is 3:1. Okay? Because for complete dominance type of gene action, these genotypes, homodominant and hetero, would have one phenotype. The same phenotype, so if you add this, 1 plus 2, 3:1. And then, since we take each gene pair independently, we have a cross of 2 heterozygous Y. We produce the same phenotypic and genotypic ratio, so that if we get the F2 of this mating, of this dihybrid cross, we have 9:3, so 3:1. How did we get this? Because the phenotypic ratio of this R here is 3:1, 3/4 and 1/4. And then, we do the branching method, no? What do we branch? Yung Y na, yung gene Y. Okay, coding for color, which is yellow and green. So, yung dominant ay yellow, yung proportion ng yellow is 3, and yung proportion ng green ay 1. To make it a fraction, we have 3/4 and 1/4. How did we get 9/16? We multiply 3/4 and 3/4, 9/16. Yun yung pagkuha ng probabilities of independent events. These two here, the R gene and the Y gene are both independent events. So how do we get the probabilities of this event or the mating? We just multiply the product of their probabilities, individual probabilities. 3/4 times 3/4, 3/4 times 1/4, and so on. We get 9/16, 3/16, and so on. How about for mutually exclusive events? Ano ba yung tinatawag natin na mutually exclusive events? Okay, when two events are said to be mutually exclusive, the probability that the individual will have one of these characteristics is equal to the sum of their probabilities. Say for example, we consider the gene Y. Okay? What is the probability that the gene Y will be inherited? What is the probability that the recessive y will be inherited. Okay? Since dalawa lang naman sila, yung probability ng dominant y ay 1/2, yung probability ng recessive y ay 1/2. So, hati sila ng probability. Okay? So that we have 1/2 plus 1/2 is equal to 1. This is the probability of the mutually exclusive events, dominant y or recessive y. Ano yung ibig sabihin nun? Ano yung interpretation nun? The probability of getting either dominant y or recessive y is 1. Why 1? You just add 1/2. With that of 1/2, you get 1. And then under this topic, we have 4, level of significance, chi-square test, binomial distribution, and normal distribution. So, gusto ko lang ipakita sa inyo yung different methods in order to determine ratios if they are consistent with the Mendelian ratios or not. Okay? So, ito ginagamit natin sa statistics. If you recall from your elementary statistics or even in your Agri 30, yung methods of agricultural research, you also have here a chi-square test, binomial distribution wherein it uses this formula and then yung normal distribution. So, to simplify things, we'll just use 1, One is yung chi-square test lang yung gagamitin natin, which is denoted by x squared. And this is equal to the sum of the squared deviations all over the expected value. Okay? This is the sum of the squared deviations all over the expected value. Wherein O refers to the observed value, E is the expected value. Okay? So, to illustrate further, we have here a sample problem, Let me read to you. Results of a phenotypic analysis of 96 F2 progenies in 2 replicate experiments are given as follows. So, we have here experiment 1 and 2, and these are the resulting phenotypes that are observed from the experiment. So, sa experiment 1, we have 70 for the phenotype 1 and then phenotype 2, 26. So, yung second experiment, we have 76 and 20. So, most probably, yung phenotype 1 yung dominant and yung phenotype 2 yung recessive based from the ratio of the observation. And then it says here, calculate the chi-square statistic for each experiment using a 3:1 ratio and then using a 13:3 ratio. Now, how do we do that? We have to, we just substitute the values or plug in the values to the given formula. Okay, let's consider first experiment number 1. We have to rearrange the table. So, for experiment number 1, we get the phenotype 1 and 2, and then we make a column for observed values, and we make a column for expected, and then we make a column for the computation of the deviations, So, for the phenotype 1, yung observed value is 70. Sa expected, how do we get, by the way, the expected values? So, ang sinasabi dito is 3:1. So, ang total nitong observe ay 96 because you add 70 plus 26, you get 96. Now, how do you get them 3:1? So, 3:1, yung 3 doon, 75%, yung 1, 25%. So, you multiply 0.75 to 70, to 96 rather, to the total, to get the expected value for phenotype 1. So, 75% of 96, that is 72. And the remaining 25% of 96, that is 24. If you add this, 96 siya. Yun yung expected value. Ano yung expected value? Yun yung value if you base it from Mendelian segregation. Okay, let's substitute these values to the formula. Observe minus expected, and then we substitute the values to the formula. Observe values minus expected and the square. So, 70 minus 72, we have and then you square it divided by the expected value, which is 72, that is 0.0555. Do it again for the second row. You get 0.1667, and then since chi-square siya, we have to get the summation or the total. Okay, so the total of this is 0.222. Okay, so the value of the chi-square statistic is 0.2222. Or you can just simplify it to 2 digits, no? 2 decimal places rather. So yun yung pagkuha ng chi-square statistic. So for your assignment, do the other ratio. 13:3 ratio for experiment 1. And then for experiment 2, you do the 3:1 ratio and 13:3 ratio also. That ends our discussion for Chapter 3, Gene Segregation and Interaction. So I hope you learned something from Chapter 3, and we'll see each other again in my next video for Chapter 4, Linkage and Recombination. I hope you learned something from my lecture videos, and thank you very much for patiently listening to my discussion. If you have questions, don't hesitate to ask me in the GC. Thank you.